Hello everyone, my name is Yuchen Shen. I'm a student from HSNU. Today I'm going to report about effects of agrobacterium rhinogenes, rolB gene, on plant growth in arabidopsis. Let us introduce agrobacterium rhinogenes. It can affect plants and cause crown gulls, as you can see in the picture on the left. And in its RI plastic, there's a set of genes named Rho genes, which can produce hairy roots. They are Rho A, Rho B, Rho C, and Rho D. In Rho A, it can affect plants' morphology and hormonal cycle. In Rho B, it's the most important genes for hairy root structure, and it's the one we are going to discuss about later. In Rho C, it can affect plant morphology and promotes production of secondary metabolites. In Rho D, it can only be presented in tDNA region, which is related to auxin. I'm doing this report because the secondary metabolites that Rho gene help produce can be an important ingredient for the medicine of cancer. Let's move on to our purposes. There are 4 purposes in this research. First, growing arabidopsis for transformation. Second, cultivate agrobacterium rhinogenes for infection. Third, construct arabidopsis expressing RhoB and YFP fusion proteins. And last, confirm the roles of RhoB and protein in arabidopsis. Let's move on to our research and methods. With the research method and processes. First, we're going to design recombinate DNA. There are 3 steps. First, making TLB gene cassette. We are going to insert 3 things, including RhoB, YFP, and herbicide-resistant gene inside arabidopsis. A herbicide-resistant gene can help us to ensure whether RhoB and YFP is existing in arabidopsis. After transferring TLB into agrobacterium rhinogenes, we will be cultivating transgenic agrobacterium rhinogenes. And later we will be doing construction of TLB plants. After cultivating agrobacterium rhinogenes, including TLB, we'll be growing arabidopsis, which after a week, after harvest, after planting, we will do herbicide screening. But first, we will be checking dilution of the concentration of test pasta. First, in the first of the experiment, we tried 0.1 of concentration. However, after the experiment, we found that most of the plants died. Therefore, we can infer that it is not an appropriate concentration for arabidopsis because we cannot choose whether this one contains the resistant gene or this one doesn't, all died. However, after the second test using 0.05, some lived and some died. From that, we can be sure it is an appropriate concentration because it can help us to check if there is resistant gene inside the arabidopsis, which means it also can include RhoB gene and YFP. So arabidopsis can be successfully infected with the concentration of pasta in 0.05 per liter. Let's move on. After basta concentration, we will do sterile planting, which means we are going to put all the arabidopsis in an environment with no bacteria. And after a week, we will test the relative survival rate. And later, we will do screening for homologs, including extracting geometric DNA and PCR. This is the result of PCR. As you can see, there are 24 squares in this picture, which means there are 24 arabidopsis. And from 1 to 9 and from 11 to 23 to 24, we can see that they are bent on the square, which means this includes the gene inside the arabidopsis, and it will be around 350 base pair. And the only one, which is number 10, is dark, which means that there is no gene and no protein inside the arabidopsis. Let's move on. After PCR and screening for our protobasis containing rolB gene, we'll do aspartic planting, which we will conjugate focus microscope to observe the action position later, which leads us to our results. After planting for a week, we did the comparison of the wild type and TLB. As you can see in the picture, the wild type and 4 kinds of TLB 3, 4, 5, 6. From this picture, we can know that there's no significant differences between the length of the main root and the amount of the side root between wild type and TLB. However, from letting it grow for months, we can see clear differences between the two. In the wild type, its stems grow upright. However, TLB transformed stems grow laterally. Next, after putting it into a fluorescein microscope, we can see there's color in the root tip, the root cap, and the meristem, which means that the YFP protein are presented there. Let's move on to conclusion. First, we successfully grown our protobasis and completed the transformation of TLB. Second, with the process of PCR, we have found plants containing rolB genes successfully. And last, we can confirm that there is performance in the root cap. In our future look, we are wondering is there any difference in the expression of rolB in the roots or plants of different sizes? And second, it is hoped that the mechanism of action of rolB can be better understood in order to apply the method of introducing hairy roots to different plants. Last, acknowledgement. Thanks to Professor Li Kunda, Department of Biotechnology, National Taiwan University, and thanks to Han Rongguo, also the Department of Biotech, National Taiwan University. That's all for my presentation. Thank you for your participation.